So, welcome back. Today, I'd like to talk very briefly about meiosis. Just going to do a quick overview of the mechanics and the functions of the process. Right? So, meiosis is a type of cell division designed specifically for the purpose of sexual reproduction. So, meiosis is what's going to make gametes, which is like sex stuff, like sperm and eggs or spores if you're a different type of organism, right? So all those little cell things that we use to make babies with come from the process of meiosis. Meiosis specifically creates daughter cells called gametes with half the ploidy of the parent cell. So for a lot of organisms like us, we are diploid, right? The gametes are haploid, one ploidy. There are other ploidies out there. So if a parent organism is 12 ploidy, their gametes will be 6 ploidy. You need half of whatever the parent has. That way when you take one, take the other, you mush them together, you get a full one, right? Half plus half, one. Right? So that's what meiosis is doing. Right? Mechanically, it is very similar to mitosis, right? So much so that we still have the same overall steps. We have prophase, metaphase, anaphase, and telophase. However, in meiosis, we're going to run it twice. So we have PMAT1 and we have PMAT2. Right? This makes sense because through mitosis, we take a 2 copy, we divide it once, and we get 1 copy, 1 copy. Right? For meiosis, we start with 2 copies. We divide it the first time, we end with 1 copy, 1 copy. We divide that again, we end with half copy, half copy, half copy, half copy, which is what our aim is, to get to half copies. Right? So we're running PMAT twice for meiosis. So in my cute little model down here, right, the organism that I've designed is a diploid organism, right, as is expressed by this right here, 2N. Right, that's how we express diploid. Right, so 2 is di, and the N represents the ploidy. Right, so N here stands for number of chromosome types. And I've defined this chromosome as having this organism as having 2 different chromosome types. Right? So for instance, humans have 23 different types. This one's more simple. It only has 2 types. So chromosome 1 has the paternal chromosome variation here. It's in black. And the maternal chromosome variation here is in red. So these 2 chromosomes are going to have exactly the same genes, right, with slight variations in the details of sequences called alleles. Right? But overarchingly, they're the same chromosomes, just slightly different. Chromosome 2 has the paternal variation that is blue in this model, and the maternal variation is green in this model. So here's our little key for what is happening along the way here. So we're going to start off cell division post S phase. So we have 2 copies of each type of chromosome. Right? So I have 2 copies of the black paternal one. I have 2 copies of the red maternal one, 2 copies of the blue paternal one, and 2 copies of the green maternal 2, right? So I've lined them up with paternal and maternal, and then maternal and paternal, to demonstrate that there's not a specific sequence here, right? One side isn't all paternal and the other all maternal. The way that they organize at the very beginning is random, through what's called independent assortment, which is very important for creating the vast randomness and different types of gametes that we can make, right? There's a very, very, very large variety of gametes that we can make because the assortment at the very beginning is random. So it could go paternal, maternal, maternal, paternal. It could go maternal, paternal, 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 maternal. Doesn't matter. Right? But each side is going to get 1 variation of each chromosome. Right? So each side will have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and the other side will have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, however many chromosomes you have. Each side is going to get 1 copy. So that is prophase effectively, right? We're doing prophase 1. We're organizing our chromosomes into chromosomes, the tight little packages that can be moved. We're breaking down the nuclear envelope. And then we move into metaphase, where we line up the chromosomes in the middle. Now, meiosis, this is a very, very important step. It's slightly different than mitosis. This is a little bit more detailed than mitosis. For meiosis, we have to line up all of the homologous chromosomes next to each other, right, down the middle of the cell. So I have chromosome 1s lined up next to each other. I have chromosome 2 lined up next to each other. Right? It would be chromosome 3. It doesn't have to be in order. It can be 1, 3, 5, 7. Right? All that matters is that number 1 is paired with number 1. The homologous chromosomes are together. This allows for a process called crossover. Crossover is when these two inner homologous chromosomes physically touch. They're touching each other, bumping into each other. Bum, 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 right? And so they have the same genes in the same region, and so they can exchange their variations of their genes. Right? So wherever they touch, they swap genes. Okay. They haven't changed what genes are there, but they've changed the allele setup. So you get these slightly different variations after crossover. And the variants are called recombinant chromosomes. Okay? So they're no longer the same as the maternal or the paternal chromosome. They're their own unique thing. And the crossover can happen anywhere along the chromosome, right? So this process creates pretty much infinite options for what our gamete genetic composition is going to be. Right? So crossover is very important for creating the diversity of genes that are made available through sexual reproduction. Right? So after metaphase happens, right, we go into anaphase 1, and we separate them onto opposite sides of the cell. Telophase 1 rebuilds the nuclear envelope temporarily. Okay. So at this point in time, we are now down to a haploid cell, right? This cell only has the paternal copy of this and the maternal copy of 2, right? So it has one copy of 1 and one copy of 2. This is now a haploid cell. It has 2 photocopies of that variation, right? But it only has the genetic information from just one variant. Yeah? Same here. This stage doesn't really have a name, right? Like this is a parent cell and these are daughter cell gametes. This is just kind of a transitionary state, right? But we are already haploid at this point. So then we move into PMAT2. Prophase breaks the nuclear envelope back down. And then metaphase lines all the chromosomes up down the middle of each of the 2 cells. These are 2 now separate areas that are being worked on, right? And so then we line these up down the middle. So if this had 10 chromosomes, it would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. We have 2, so it's 1, 2. And then we have 1, 2. Okay. Then we separate them. We split the sister chromatids apart. Right? So these are sister chromatids. We grab them and we rip them apart, pluck, pluck, right? And we move them to different sides of the cell. So we end up with one variant here, one here, one here, and one here. So this is this side is this is this. Ah, I broke it. Stop. Oh no. It's all over now. It's never going to be the same. I can spin this around, spin, 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 spin. It's never going to be the same now. It's all ruined. Okay, so anyways, this went here and this went here. In the other cell, in the other area that we're working in, we have this goes here, this goes here, this goes here, this goes here, and this ends up with our gametes. Okay, we have one copy of one variation of the chromosome. And so this gamete will have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, one copy of each. This gamete will have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, one copy of each, so forth and so on. Now, due to independent assortment and random chance of crossover, every time we go through meiosis, we're going to get different alignments of gametes, right? So that's where all the genetic diversity comes from, is this random assortment stuff going on in meiosis. And then, so like if I was a dude, which I am a dude, last time I checked, these would be spermies, right? So here we go, we make a sperm, right? And then these sperms would meet up with an egg somewhere over here, right? And the egg would have half the information as well. and then we could make babies. And then we'd have a full diploid. Baby. Right back to being an organism. Yep. So yeah, that's it. Meiosis for making gametes to make babies through sexual reproduction. PMAT1, PMAT2. Go from being diploid to haploid in most organisms, including humans. Cool? I think that's it. Awesome. Have yourselves a most fantastic day. Bye.